So this is the first one. It's of course. First of all, I want to thank the organizers for inviting me for this distinguished meeting. Could I have some light? Sorry. Really sorry. Today, ladies and gentlemen, as you all know, life on Earth appears to be adapted mainly to moderate temperatures, for example 10 to 40 degrees centigrade. This is also true for the smallest living beings, that is the bacteria. A few heat-loving organisms have been known since a long time. Already in 1862, it had been Ferdinand Kohn who had reported of green layers of cyanobacteria which grew at 55 degrees within the hot springs at Carlsbad, Czech Republic. 110 years later, in 1972, James Bradley and Tom Brock had discovered sulfur lupus, a microbe thriving at 75 degrees centigrade within the hot acidic springs at Yellowstone Park National Park. At that time, sulfur lupus had been seen as a kind of curiosity, growing at such extremely high temperature with such a hot lifestyle, which seemed to be only possible by the strictly anaerobic metabolism of this organism. It seemed to be a kind of prerequisite to cope against all the destructive forces of heat for this organism. Now, Louis Pasteur had observed bacteria of the human environment that they are safely killed by heat treatment at 80 degrees and above. The pasteurization technology is based on this finding. On Earth, the first traces of life had been, as you all know, discovered within 3.8 billion years old remains of cratons, for example, at Pilbara, Australia, and these were at Greenland. Well, that's what the early Earth may have looked like at that time, 3.8 billion years ago. Of course, nobody could take this picture at that time. I have got it from Chris McKay at NASA as a reconstruction. As you may see here, it had been a paradise of microbes, a hot, dirty, humid world, generally anaerobic, with a lot of volcanism, and still, at the end of the Great Bombardment, serious impacts which brought in a lot of heat. Only superheat lovers, similar to the hyperthermophiles existing on today's Earth, should have been able to thrive and survive in such an early Earth scenario and may have become our early ancestors. At Regensburg University, I have a strain collection of about 1,500 hyperthermophiles. Among those, even isolates which survive autoclaving for hours at 121 degrees centigrade. In my today's lecture, I want to focus mainly on the hyperthermophilic archaea, which grow up to the stability limits of the building blocks of life. As you will see later within the tree of life, these prokaryotic microbes cover all the deepest and short branches, indicating that hyperthermophily had been a very ancient feature in the history of life. My hunting for novel archaea began in 1979, shortly after the discovery of the archaeal third domain by Carl Woese. On my first trip to Iceland in 1980, together with the world-famous molecular biologist Wolfram Zillig, I found some really exciting hot environments. Here's Wolfram, and so these were really boiling sulfuric mountains in the Krafler area. This picture had been taken by myself 3 days before a fissure eruption had occurred on this place. And hot, anaerobic, as we found out, mud was thrown up into the air, and steam, hydrogen, carbon dioxide, and H2S and some carbon monoxide, I think, because our heads on some places turned really red, and we got a lot of headaches. This must have been the CO occurred there. We had in our backpack a field microscope, and we found huge antler-shaped microbes which had never been cultured before within this thrown up mud, and they were up to 10 to the 7 cells per milliliter. It's almost like in a bioreactor, and so we were very surprised because this was strictly anaerobic, as we found out by adding resveratrol, which immediately turned colorless. In the lab, later, we were able to grow these organisms. They were strictly anaerobic, growing up to 97 degrees. Nobody has expected such a thing at that time. And we named them, together with Wolfram and myself, the Thermoproteales. Well, the question was, could there exist organisms even thriving above 100 degrees centigrade within superheated water? Well, it's a seafloor of Vulcano, close to the Vulcano island. It was able to go by foot down by scuba diving, and I was scuba diving there already in 1981. And so there were, depending on the heat of the volcano, there was superheated water with temperatures up to 105 degrees. And I took some samples there, and then in the laboratory, these samples under overpressure and strictly anaerobic conditions gave rise to a novel organism which was able to grow up to 110 degrees. I almost couldn't believe it. I always thought maybe there is some lab artifact, but it was really true, and I named it Hyperthermophiles occultum. So this is this culture bottle. It was pressurized by hydrogen and carbon dioxide, and here was because at the bottom where I took these samples, there were a lot of sulfur crusts which had formed by oxidation of the H2S coming out, and so I added sulfur, and you see here possibly the cobwebs covering the sulfur, and those formed overnight at 105 degrees. This is the experiment, and this is a high intensity illumination, the light microscope, and you can see cells connected by a network of so far unknown organisms. That's how it looked like in the electron microscope, this pyrodictyl. These were networks of hollow tubules, so-called cannulae, made up of proteins stable up to 140 degrees. And this organism demonstrated for the first time that life exists at least up to 110 degrees on our planet. With this experience in mind, during the following years, I visited high temperature areas all over the world and isolated high temperature organisms from there. For example, from deep sea environments. My friend Holger Janasch from Woods Hole was kind enough to invite me to go with him and use his equipment. And there's wonderful deep dive submersible Alvin. This was off the coast of Mexico at 21 degrees north, a depth of 2.5 kilometers, and hot water is escaping. And as I found out, so these are porous chimneys, and the venting hot water is 300 to 400 degrees, much too hot, of course, would be more than life in the frying pan. There is no life if you take serious samples here, careful samples, but these, like huge macaronis, the walls of the black smokers, they are porous, and they suck in also water from outside. Outside is 2.5 degrees or whatever, and inside 300 to 400 is the central heating, enormous temperature gradients, and this is the place where the hyperthermophiles are really living, and not only hyperthermophiles, of course, but also from seemingly unsuitable places. I was able to isolate hyperthermophiles, like within the bed of the North Sea, or here at the North Slope in North Alaska. It's a Prudhoe Bay, and I took samples of the oil stratifications there. The oil field are situated in a depth of 3 or 4 kilometers. There's about 400 meters permafrost soil, almost reminding a little bit on the planet Mars, but deeper below, also there is no volcanism, it turns hot, and the upcoming oil water gas samples are still, mixtures are still hot, and also down there is sand and limestone. And back in the lab, highly unexpected at that time, but I hoped so that there may be something down, because there was always strange H2S was formed that was all attached to chemical reactions there, because it was much too hot that they thought these oil people, that organisms could grow there. And this was the first time we had found this. It was an overwhelming variety of hyperthermophiles there, as you see in this table, and unbelievable, the upcoming cells, so forget about these names, I come to this later, but the upcoming cells were up to 10 to the 4th to 10,000 per milliliter, which just had been released from these communities deep down there in North Alaska, and in this case it was in System Alpha platform I had visited down under the bed, 3 kilometers down under the bed of the North Sea, where it's also no volcanism, but of course geothermal heating. In general, the samples, this was from my lab, my people and I brought the samples back from different environments to the laboratory, and there we did a lot of growth experiments and characterized and described the organisms, and I'm happy that many people are still working with our microbes in different fields of science and even biotechnology. And just in a nutshell, here's some examples of the newly found genera of hyperthermophiles. This is Thermotoga, a bacterium growing up to 90 degrees. It's a heterotroph. This is Aquifex, the water maker, I call it, a microaerophile growing by hydrogen oxidation, but it grows at hydrogen concentration as low as 10 ppm, even in the laboratory. Let's go on. This is Acidicanus, a thermoacidophile, which grows in very acidic environments. It's able, depending on the redox potential, either to oxidize or to reduce sulfur. The Thermoproteales, Staphylococcus, a heterotrophic hyperthermophile, Pyrococcus furiosus, the Russian fireball, which grows fastest at 100 degrees, a doubling time of about 20 minutes, almost like an E. coli at 37 degrees. Archaeofluorus, the first archaeosulfate reducer, or Methanopyrus, a methanogen growing, this is Methanopyrus candleri, which I named it, an organism which grows up to 110 degrees with a temperature optimum of 100 degrees, showing for the first time that biogenic methanogenesis is possible at temperatures above 100 degrees. Well, from other people have described 8 or 10 years later the same, they found a similar organism, it's even the same species, and said it grows up to 121 degrees. And this was published. But as I was able to get the strain, and we checked it in our laboratory, and it seemed to be more possibly a problem of calibrated thermometers or so, because we found exactly the same values as we had for our organism. It survives for several hours at 121 degrees. The Hyperthermophiles can't, our Hyperthermophiles can't really, but the other one is not different by the upper temperature limit. From a black beehive smoker at the mid-Atlantic ridge in a depth of 4 kilometers, I took samples and isolated Hyrolobus fumarii, the organism with the highest growth temperature in our hands so far. This organism is unable to grow at 90 degrees centigrade because it's still too cold, and it begins slowly to grow above 90 degrees, as you may see here. The doubling time versus the temperature, you see the optimum is around between 100 and 105 degrees. The maximum, for unknown reasons only, it's a pretty sharp growth ranges in our hands up to 110 degrees, survives for hours. Autoclaving, also this organism is unable to make spores. So the vegetative cells, they are so sturdy. Also in this case, another isolate had been found, which is very similar. And unfortunately, it says that it grows at 120 degrees. This trait is not available at all for science. It doesn't exist. Just we tried to reproduce this data. It would have been very nice to have an organism growing at the autoclave, but not so far. It's just survival. I could comment on this also later. Well, in their physiological growth requirements, like temperature, pH, ionic strength, and redox potential, and also the energy sources, hyperthermophiles are specifically adapted to the hot environments, and all cell components have to be heat stable. Well, most hyperthermophiles are strictly anaerobic, as we found out, so sulfur lobus is really an exception, and they require anaerobic culture conditions. At ambient temperatures, however, hyperthermophiles are able to survive for years, or I can say now since 20 years, 25 years, I have very old samples still, and they give rise to the organisms. We do it regularly, or my successor in Regensburg does in the lab course. So, and they survive even in the presence of oxygen. If it's cold, but if it's hot, they are killed within half an hour, or even less. Well, many of these hyperthermophiles are strict or facultative chemolith autotrophs using inorganic energy and carbon sources, and hydrogen, as Professor Bedonkin had already mentioned or discussed in detail yesterday. Hydrogen is a very powerful energy source also for hyperthermophiles, for methane, magnetite, and hydrogen sulfide, or even water. And in addition, they need these are chemolith autotrophs. They require just heat, trace minerals, and liquid water. Well, in Moses' small subunit RNA based tree of life, without any exception, hyperthermophiles cover all the deepest and short lineages. As you know, this is a bacterial domain, as Carlos called it, the archaeal domain. These are the eukaryotes. Within the eukaryotes, for unknown reasons, there are no hyperthermophiles. Well, this is not the origin of life, of course. The origin of life would be 3 floors below. This is the last common ancestor, because this is all based on the ribosomal RNA. So the organism had to have already at least a precursor type of the ribosome. And there are the nanoarchaeota also, and there's a question mark because this is an organism, the nanoarchaeota, which are so deeply branching that the branching point is still unclear. The same is true for the core archaeota. Now, let me discuss just the nanoarchaeotes a little bit in more detail. The first representatives, which are named Nanoarchaeum equidans, the riding little dwarf archaea, have been cultivated from the Koldensee hot submarine vents north of Iceland, and it requires obligatory a host to grow, and the host is Ignicoccus hospitalis. Oops. Oh, here. So, and the nanoarchaea, there is a virus-sized organism, very, very small, and can easily overlooked in the light microscope. It's almost invisible because there's only a diameter of 0.4 micrometers here. Oops, sorry. Here you can see the host, the Ignicoccus and Nanoarchaeum. These are the 0.4 micrometer cell sitting outside. This is an enlargement of the attachment site. And so Nanoarchaeum is unable to grow by itself, but it requires obligatory the Ignicoccus, while the Ignicoccus host is a chemolitho autotroph growing on hydrogen by sulfur reduction, and a strict chemolitho autotroph. It's able to grow also alone without the nanoarchaea in the laboratory. The genomes of both organisms have been sequenced by Narversa, and with only 491 kilobase pairs, the Nanoarchaeum equidans genome is amongst the smallest of a living being. So why is it really not a virus? The reason is that it clearly has membranes, and it has its own ribosomes, which you can possibly see here. Here's the membrane and the ribosome. So it's clearly an organism, a living organism. Well, the extremely small genome size could be due to genome reduction, which is common in parasitic bacteria. In consequence, as a rule, parasitic bacteria harbor hundreds of non-functional pseudogenes. In Nanoarchaeum, however, also whole groups of genes which are essential for independent life are missing. As you may see here, here are regular Hyperthermophiles, archaea, and this is Nanoarchaeum, and the different groups of corks. And as you may see here in green, the metabolic genes in Nanoarchaeum are almost completely missing. And so the question is, how is it with Nanoarchaeum? Does it have pseudogenes? And there are not one pseudogenes. They have no pseudogenes. That's genes which were degrading when they became parasites. So, and this is very much in contrast to the bacterial parasites. Also, they have no living relatives. Therefore, if at all, a gene reduction in Nanoarchaeum may have occurred very early in the history of this organism. Alternatively, the Nano-Ignicoccus system could even be remains of very early pre-Darwinian communities as postulated earlier by Rose and Cantor. At present, we are far away from a deeper understanding of the Nanoarchaeum Ignicoccus system. There are still very highly unusual features as recently shown by my former Regensburg lab, Ignicoccus exhibits a cellular organization so far unique to all living organisms. So this is a 3D reconstruction so this organism possesses two membranes. The outer is the cellular membrane, which harbors the ATP synthesis system. But in addition, there's a further inner membrane, which you can see here, with many fragments budding off, and this inner membrane surrounds the information storage and processing system. That means the DNA and the ribosomes. Well, these nanoarchaeotes are no curiosity. They are distributed worldwide within terrestrial and submarine hot environments, and they have been completely overlooked before their first cultivation. Finally, maybe a few words about the core archaeota. The unique ribosomal RNA of the core archaeota had been discovered in the pioneering work by Barnes and Monopase already in 1996. And this was all which was known, the environmental 16S RNA of this organism, and we were able to culture them, these organisms, and by cultivation we found that really small, 0.16 micrometers in diameter. This is an electron micrograph, so they're all practically invisible in the light microscope, and this was the reason why people had overlooked them. And they are kind of needles, 10 to 100 micrometers long, with blunt ends, and they have a unique S layer, as you see on the right side. Well, in collaboration with JGI, we were able to sequence and annotate the core archaeum cryptophyllum genome. It has only a small genome, but not very tiny. It's 1.589 kilobase pairs, rather small, about 3 times the genome of Nanoarchaeum. Based on its ribosomal RNA sequences, Archaeum cryptophyllum represents the deepest branch within the Kren archaeota. And however, when we analyzed other genes of this organism, it revealed an unprecedented combination of genes which had been thought to be characteristic either to the Uri archaeota or to the Kren archaeota, a hybrid of two kinds of genes which had been taken as signature genes for these different kingdoms of archaea. Well, here just an example of genes which are either, which had been thought to be signature genes of Uri archaeota and others of the Kren archaeota. So this had not been expected at all before. Well, the heterogeneous gene complement suggests that the core archaeota diverged from the two major archaeal lineages, two kingdoms, very early, and further comparisons of these unique organisms may illuminate the early evolution of the archaea and the nature with its shared ancestor, with the eukaryote. Certainly, these findings are only the tip of a very hot iceberg. Organisms with similar properties may exist also on other planets and moons. Whenever volcanism and liquid water come together, at least within our solar systems, they could have been distributed by impacts already at the end of the Great Bombardment. And I'm pretty sure that Lisa, our next speaker, will be able to culture them. Thank you very much for attention. Elizabeth Blöck, Chip Elkins, Michael Horn, Harald Huber, and Brenda Brown, and we were still in collaboration with Mark Adams, Jorge Janosch, Milton Koda, Paul Richardson, and Gerhard Wanner. Thanks.
Thank you.